Hello, my name is Brian Hung. I'm a rising senior at the University of Oregon, majoring in human physiology with minors in bioengineering and chemistry. This past summer, I have the privilege to work in Dr. Darren Baker's lab at the Mayo Clinic here in Rochester, Minnesota, with NIH SenNet CUSP summer internship. My project was specifically looking at naturally aged spinal cord and its relation to senescent cell burden. With that said, here's the age mice exhibit signs of increased senescent cell burden in the spinal cord before signs of nerve cell loss. Before we dig in, I think it's important to establish what aging means in this case. Aging is the time-related degradation of physiological function essential to survival and reproduction. And oftentimes, aging comes with various type of anatomical and physiological changes within the organism. A lot of studies out there have shown that many age-related chronic diseases are highly linked to the accumulation of senescent cells. What is senescent cells? Senescent cells are when a normal cycling cells enter what we call a cellular senescence. That is when cells enter a state of cell cycle rest while remaining metabolically active. And this can due to a lot of reasoning, such as organelle stress, DNA damage, telomere dysfunction. A lot of time, senescent cells are characterized by increased A beta gal activity, increased secretion of SAS factor, and one of the most common one is increased expression of the P16 and P21 protein. Through this project, I wish to assess senescent cells burden in the spinal cord during normative aging, and in hoping to further understand the relationship that maybe it can potentially help discovering future treatment for age-related neurodegenerative degenerative diseases. So here's the project overview. Our sample was young mice, which were about 2 months old, middle-aged mice, 10-month old, and old mice, that's 18 to 20 months old. We did a series of functional assessment, which include grip strength tests and hyaline clasping assessment. Both of those tests were to see if there's any muscular deficit that could potentially relate to a spinal cord deficit as well. Post the functional testing, we then did PBS perfusion to extract the spinal cord from our mice sample. Spinal cord was extracted and divided by its region. So three regions, which are cervical, thoracic, and lumbar. Within each region, the top half were used and preserved to do histology, and the bottom half was used for Western blotting. In this specific project, thoracic spinal cord was the region that I specifically look into. And there are two considerations to that. One is a technical, because thoracic tends to be the longest and the larger region within the spinal cord. And second is that with previous data from the lab, a lot of them came from the thoracic spinal cord. So here we have the first functional test, that is hyaline clasping assessment. So typically in a normal healthy mice, when you suspend the mice by its tail, we can see the hind limb spread across away from the body line, and typically more than 45 degrees angle away from the body line. And here, hyaline clasp is scored in 5 different criteria from 0 to 4, with 0 being hyaline away from the body line, more than 45-degree angle, 1 being away from the body line as well, but less than 45-degree angle, and 2 being completely parallel to the body line, 3 and 4 being within the body line, and 4 being within the body line with the legs touching. And here with the data, we can see that all mice tend to exhibit a 1 or a 2, which are significantly higher than what we observe in the young mice. And this had led us to question, could this muscular deficit be related to age-related spinal cord degeneration? So then we further continue with our second functional test, which is examining the mice's or the mouse for them grip strength. And on the left here, we have how the grip strength meter is set up, which is situated in the vertical position. The mouse will then handle by its tail and then go up to the triangle grip bar, pull down gently, which the meter will then demonstrate a negative value, indicate the mouse's forelimb grip force. Here on the right, we have a statistical graph indicating the grip strength that was performed in this series of testing. During the time period, the testing was done in a total of 4 days, with the first 3 days being training days and the last day being the actual testing days. And here we can see that all mice have scored consistently lower score compared to young and middle-aged mice. And this further also validate our thinking and question of, could this be a muscular deficit that were caused by any sort of spinal cord aging or any dysfunction? So then moving forward with the thought, we then did H&E staining using the OCT embedded tissue. And I think here it's important to mention real quick about the general spinal cord anatomy. So here in spinal cord, we have two dorsal horn and two ventral horn. Dorsal horn is where signal tends to get sent in from the peripheral nervous system, where ventral horn is where signal gets sent out to perform those functional or muscular function or any sort of physiological function. And here in the histology slides, we can see a butterfly structure in the middle of each picture, and that is what we call the gray matter, where our neurons or nuclei resides, where compared to the pinker outside area, that is what we call the white matter, where the axons and myelin tend to reside. So with that previous thought from the functioning test, we then look at the cell density in the gray matter, trying to see if there any sort of relation to decrease in neuron cells or cell density within the gray matter. And surprisingly, we didn't find any significant difference between the young mice and the old mice, which led us to think that could this be a ventral horn motor neuron problem, since we're seeing those deficits in more of a motor side of function. So then we counted the ventral horn motor neuron. And here, motor neuron are defined as the big, dark purple cell nuclei that we can observe in the gray matter. And again, we didn't see any significant changes or difference between the young and the old mice in terms of motor neuron in the ventral horn. So then we furthermore took another approach in looking at NeuN. NeuN is a neuron-specific nucleic protein. Theoretically, you would expect seeing more NeuN activity in the young mice compared to the old mice, but here we're getting the result of seeing more NeuN expression compared to the young mice. And that did shock us by surprise. And I think moving forward the project, this will be a good point to track with and follow up with. Following that thought, we then take another approach in looking at expression of senescence associated proteins. So we perform Western blotting on the common senescence associated protein, which are P16 and P21 proteins. Here, it does tend to follow what we believe would observe in old mice, which is increased expression of P16 and P21 protein. One thing that did caught us by surprise was the sex differences that were observed here, especially in the P16 protein, where we see more P16 expression in the old female in comparison to the old male mice. And that was something that led us to think, could aging spinal cord be also a sex difference when it comes to the topic of senescence? To further validate our thoughts in terms of spinal cord aging and senescence associated proteins and all the other characteristics, we then also perform A beta gal staining on the spinal cord tissue. Here on the young mice, we see a relatively low staining of blue cells, which blue cells here are indicate SA beta gal positive cells, where on the right here we see the old, which we can see there's an increase of threefold, and that further validate our thought and hypothesis that there is an increased SA beta gal activity in the old mice. So in conclusion, that we can say aged mice exhibit more severe hyaline classing compared to young mice, as well as a lower forelimb grip strength compared to young mice. We can also say that aged mice do tend to express higher senescence related protein and characteristic, but this tends to occur before any indication of nerve cell loss. For future direction, I think it'll be worth it to investigate the effect of senescent cell accumulation on various aspects of neurobiology, such as neuron cell function in the aging spinal cord. With my time here, I didn't have enough time, but If I did, I wish to perform immunofluorescence for the senescent associated marker in the H spinal cord tissue. Furthermore, I think moving outside of the central nervous system and explore more of a peripheral nervous system would also be a valuable knowledge and information for the project moving forward, especially looking at astrocyte microglia and maybe going more downstream to see if those muscular deficit or the phenotype that we're observing in the H mice could be something more downstream rather than coming from spinal cord itself. And lastly, assess differences in total gray matter and white matter area in the spinal cord region, as well as tagging along the related sex difference in the spinal cord region could also be an interesting aspect to look at in the future. With that, I would like to express my gratitude to the amazing individuals at the Baker Laboratory, especially Dr. Baker, Sarah Graves, Karthik Jagannathan, Bennett Childs, for all the amazing help and guidance, as well as the NIH SenNet CUSP Summer Internship Program and all that made it happen. Through this program, I really found self-growth from very first week till now, not even just in terms of laboratory skills or any scientific investigation, but also personal growth in terms of how to collaborate with people from different expertise and different professionals, and how to really verbally present myself and present my work and work in an environment where not everybody necessarily have the same standpoint and idea, and how to collaborate through that sort of obstacle. Uh, it really was a very memorable experience that I have, and I'm truly grateful for my time here and anything that may come my way from this experience in the future. Thank you.